Hello students, welcome to EPG Pathshala. I am Dr. Shaukat Mir from Hamdard University, Delhi. Today we are going to talk about a module evaluation of herbal drugs based on physical properties, which is under the paper Pharmaceutical and Biological Analysis. After completing this module, we should be able to understand quality control of herbal drugs, why is it important, and what are the guidelines for quality control of herbal drugs and herbal medicinal products. Parameters for the evaluation of the herbal products are also to be understood under this evaluation, under this module. We will also try to understand the challenges and the bottlenecks that we face while using physical evaluation for the assessment of quality of natural products. We will also at the end look at the future prospects and the scope of physical evaluation in the analysis of natural products. Let's first understand what is quality. So quality is a term which we know, which we understand, which we listen more often than not. So actually when we talk about quality, we actually want to know what is present and how much is present. So once a scientist wants to know what is present inside a drug and how much of that content is there, we are actually talking about the quality. So quality is a measure of content and purity of a substance. And the methods that are used to assess these quality parameters, this is known as evaluation. So quality control is a set of procedures that ensure that a manufactured herbal product adheres to the defined set of quality criteria of efficacy, safety, and potency. Another related term for quality control is standardization. As you understand that standardization represents a framework to which all relevant parties and stakeholders agree to adhere so that all the processes associated with the creation of a product or a herbal medicinal product are performed within these set guidelines. The quality control parameters for the herbal medicinal products, as you can see, there are different parameters by which the quality of herbal products can be ascertained. To name a few, we have botanical evaluation, physical evaluation, chemical evaluation, biological evaluation, and a preliminary type of evaluation consisting of organoleptic evaluation. So different parameters for under each of those quality control parameters have been mentioned in this diagram. We are today going to discuss physical evaluation, under which different parameters like moisture content, extractive values, ash values, etc. It also includes some of those spectroscopical analysis which are based on spectroscopy. So those will be dealt in separate. This slide gives the detailed information about some of those physical evaluation parameters that are applicable to herbal medicinal products. They include moisture content, extractive values, melting points, refractive index, viscosity, and others shown in this slide. So these are those properties which are associated with isolated compounds or specific compounds of herbal medicinal products. So physical evaluation is a type of evaluation where the components of the herbal medicinal product or a substance, they are not changed for the purpose of analysis. So we will be discussing all of those one by one. I want to mention again that spectroscopical analysis, which is the part of physical evaluation, will be dealt separately in another module. Now let us discuss the first parameter for physical evaluation. The first parameter is moisture content. We understand that the water content in herbal drugs in excess to the limit prescribed by the pharmacopoeias is both uneconomical as well as it starts the decay process or the deterioration of the product. So moisture content may lead to activation of enzymes as well as the growth of microbes. It also is helpful for the growth of molds and mites, so which results in the decomposition of herbal drugs. So the attack of these molds, mites, and insects gets increased by many folds if it has more of this moisture content. So because of these reasons that more of the moisture in the drug is uneconomical for transport and storage, as well as it starts an early deterioration process. The measurement and accurate measurement of moisture content is very basic parameter prescribed by guidelines of quality control for herbal medicinal products. Moisture content can be determined by the use of various methods like direct drying, chemical methods, thermogravimetric methods, or even gas chromatographic techniques. So now let's first discuss loss on drying. So this is the method where we use direct heating of the sample. So one of the most commonly used method for the determination of moisture is direct drying. What we do here is that a sample, either in the crude form or an isolated compound or a pure compound, it is heated directly at 105 degrees Celsius in an oven to a constant weight. Then the weight difference after drying is compared to the weight of the sample before drying. The method is particularly useful for herbal materials which contain very little volatile oil. Such examples are digitalis, starch, aloe, and fibers. So these are the substances. They contain lesser quantity of volatile oil, but the moisture content determination is very important for them. So a direct drying method can be used for determination of loss on drying. So this method of determination of loss of drying is recommended by all major pharmacopoeias like Indian Pharmacopoeia, European Pharmacopoeia, British Pharmacopoeia, or the United States Pharmacopoeia. So it has also been taken up by the Department of Ayush to be covered under various pharmacopoeias like Unani Pharmacopoeia and the Ayurvedic Pharmacopoeia of India. So the usually a good idea will be to have an instrument which can dry the sample at the same time as well as record its weight. This is the sample. This is such an instrument which is known as moisture balance, which has the provision of drying and recording the weight of the tested sample simultaneously. Now let's consider those samples which contain more of the volatile oil contents. So usually when you dry the sample by direct heating, so loss of weight is mainly due to water, but in samples which contain more of volatile oil, the loss of weight or loss on drying is also associated with the loss of volatile content. So in this case, for such substances which are oil containing substances, you cannot utilize direct heating as a method to determine the loss on drying. What we do here is the drying is accomplished by spreading the sample on thin layers. So the known quantity of sample is placed over the glass plates, and it is spread, and then these plates, they are put inside a desiccator over phosphorus pentoxide. So drying can be accelerated by applying vacuum also. Now let's go to the next method for the estimation of moisture content. So this is a chemical method which is commonly known as Karl-Fischer procedure. It is widely used in different pharmaceutical, chemical, and petrochemical industries. It is also prescribed by all major pharmacopoeias for the evaluation of expensive and moisture-sensitive drugs like alkaloid-containing drugs. digitalis, ipecac, alginates, as well as fixed oils that are used for parenteral preparations. The crude drug samples that are to be examined by chemical method, they are washed by anhydrous dioxane. The allocates are then titrated against the reagent consisting of iodine, sulfur dioxide, pyridine in dry methanol. This reagent mixture is usually brown in color. So with the addition of this, the allocates of containing the moisture, the color disappears for some time, but as this water content is titrated or it is exhausted, so it retains its brown color of the reagent. So the end color is shown by the retention of dark brown color in this titration. The reaction that is involved in this method has been shown in the lower panel. So we see that water, dry methanol, iodine and sulfur, when pyridine is also there, so it results in the formation of pyridine sulfur trioxide followed by the pyridine salt of methyl sulfate. So as here you can see the R represents pyridine. So there are also images of Carl Fischer apparatus that is a common feature in all analytical labs. This method is thermogravimetric analysis. So here the method relies on the separation of water followed by its determination. So usually distillation is used for the separation of moisture from crude drugs and foodstuffs. Then what the distillation is carried out by water saturated organic solvents like xylene, toluene, carbon tetrachloride. Then the moisture in the sample co-distills with this organic solvent and it's condensed and is collected in Dean Stark trap. So this mixture of organic and water, organic solvents, it separates into two layers and the content of moisture can be directly read from the graduated tube. The other important method that is used for the determination of moisture in crude drugs is gas chromatography method. Here the sample which is to be analyzed for moisture content is extracted with dry methanol and these allocates, they are injected onto the column consisting of Borovex on fluoropack or Poropack. The components, they get separated and are recorded in the form of a chromatogram. The content of water in the weighed samples, in the already weighed samples, can be readily read out from the resulting chromatogram. So this method is used extensively nowadays for the determination of moisture content. Now coming to the next parameter, which consists of extractive values. So this is a very significant and preliminary type of physical evaluation method that is used for crude drugs. It is particularly preferred when the other methods or parameters cannot be applied. So here you can go for the cold extractive values, hot extractive values, and the successive extractive values. So cold extraction, there is maceration with different organic solvents, while in hot extraction, the Soxhlet apparatus is used for the determination of extractive values, while as in successive solvents, they are changed with respect to their polarity from low polar to more polar solvents. The total content that is obtained using a solvent, be it in cold extraction, hot extraction, or successive extraction, so it can be calculated as a ratio of weight of the residue in grams to the initial weight of the sample taken in these experiments. This slide shows water-soluble extractive value and alcohol-soluble extract value procedures. So they have been discussed separately because many of those pharmacopoeias, they prescribe percentage or the limits of what are these soluble or insoluble extractives as a parameter for quality evaluation. So as we can see that alcohol soluble extractive value, so a sample is coarsely powdered and then it is extracted with alcohol for 24 hours. For first 6 hours it is intermittently shaken and allowed to stand for next for 18 hours. The sample that is collected is evaporated to dryness, and this extract, that the residue that you get, it makes the alcohol soluble extractive. While as in water soluble extractive, sample is again taken in the powdered form and it is extracted with water, and then following the procedure as depicted here, we can continue to dry it to a final residue. The weight of the final residue to the initial weight of the sample can be used for the calculation of water soluble extract. In the table we can see that in gentian and glycyrrhiza, percentage of water soluble extract is used for its quality evaluation, while as for Coelia, valerian and ginger, percentage extractive to different strengths of alcohol, ethanol, they are prescribed, while as in the third one, in other cases, there are limits for ethanol insoluble and limits for petroleum soluble extracts. Third parameter for physical evaluation is volatile oil content determination. So as these are applicable to volatile oil containing drugs, it is official requirement for many of those drugs. So here a Clevenger apparatus shown on the right hand side, they are used. So A is the Clevenger apparatus that is used for oils lighter than water, while as B is the Clevenger apparatus that is used for volatile oils heavier than water. So the process of distillation is followed. Oil and water condenses in the graduated tube, and the excess of water that is circulated back to the system, while as the content of oil can be read directly from the graduated tube. Next set of parameters under physical evaluation consist of ash value. So we have different types of ash values like total ash, acid insoluble ash and water insoluble ash. In addition to this, we have sulfated ash also, which is recorded at more higher temperature. So in these samples, in these experiments, the sample of the crude drug, these purified compounds, the samples are incinerated in silica crucible at a temperature about 450 degrees Celsius so as to make it carbon free, and then the total ash is measured. While as in case of acid insoluble content, the total ash is boiled with 25 ml of dilute HCl for 5 minutes. The insoluble matter is filtered on ashless filter paper, washed with hot water, and again heated at 450 degrees Celsius to get acid insoluble ash. Same is the case with water soluble, where the ash is treated with water and the water soluble content of the ash is determined. Next parameter is refractive index. So we understand that there are some of those chemical compounds and natural substances which are their values of refractive index. So actually this refractive index is the ratio of the velocity of light in vacuum to the velocity in substance, and it is represented as a ratio of sine of angle of incidence to the sine of angle of refraction. So refractometers are used for the determination of refractive index. The refractive index of some of those oils are given in the lower panel. You can see the cassia cinnamon oil has a refractive index of 1.6. Cinnamon oil is 1.53, and arachis oil and castor oil values, they are also there. We have values of refractive index ranging from 1.52 to 1.53 for clove oil. So these oils, their quality is measured with respect to the determination of their refractive index. Next parameter for the evaluation of natural products and herbal products is optical rotation. We understand that many of those natural compounds or these products, they have the tendency of rotating the polarized light passing through it either to clockwise or anti-clockwise. The clockwise rotating substances are known as dextrorotatory, while as anti-clockwise rotating substances are known as levorotatory substances. We have values for optical rotation of many of those isolated compounds, as well as products like honey, caraway oil, clove oil. As mentioned here in this slide, you can see that lemon oil has a value of plus 57 degrees to 70 degrees, We have cinnamon oil, which is levorotatory for either 0 to minus 2 degree values. Spearmint oil in the same way, it is very strongly levorotatory. Next physical evaluation parameter is melting point. So it is very important for the evaluation on the basis of physical property. Here melting point is used as the method of evaluation. Here we use finely powdered substance, so which is transferred to a dry capillary by tapping it to a by tapping it. So the capillary which is fused on one end, it is filled to the level of 6 to or about 6 mm. So then it is placed next to a thermometer inserted inside a melting point apparatus. So the melting point apparatus has provisions for inserting a thermometer as well as a capillary so that both are visible at the same time. So thermometer is placed in a way so that the graduations are very, very clear. So it has illumination inside, so we can see the graduations of the thermometer as well as the physical state of the substance in the capillary. So it is slowly heated and it is watched closely so that the point so that the point at which the liquefaction of the substance takes place, we record the temperature in the thermometer and record the melting point. We have specific gravity as another parameter for the quality evaluation on the basis of physical properties. So as we understand that specific gravity is the ratio of the density of an object and a reference substance, which is generally water, at a specified temperature. So we have substances heavier than water or lighter than water. So accordingly, we have specific gravity, which is a ratio of density of liquid to the density of water. So we can determine the specific gravity of substances using an hydrometer or a specific gravity bottle as shown in this diagram. Another important parameter is viscosity. Viscosity is one of the important physical characteristics of volatile oils and fixed oils. So all these liquid samples, they can be evaluated. If they are viscous, they can be evaluated with respect to their viscosity. Viscosity is defined as the resistance to flow and shear. So any type of shear that is applied on these oils or liquids, so resistance offered by the resistance offered by these substances or liquids, so it is measured with respect to viscosity. So this is a measure of its flowability, pourability, how easily it can be poured. Viscosity of a liquid is constant at certain temperature, and so when you record the viscosity, the temperature needs to be maintained. Mostly viscosity is measured at two temperatures. So the industrial products, the industrial oils, they are the viscosity for them is measured as at higher temperatures, somewhere like 40 degrees Celsius, because their use is on a higher temperature basis. So their grading system and they are to be graded as per ISO viscosity grading system. While as the pharmaceutical products, they are to be used in low temperature conditions, not going to the level of 40 degrees Celsius, especially in exception to those used in the African regions. While as for the measurement of the viscosity, we use two types of apparatus which are known as viscometers. We have rotary viscometer and capillary tube viscometers. So rotary viscometer has the spindles. So these spindles, they are rotated on a surface of a liquid, so the shear or the resistance offered by gets recorded, while as in capillary tube viscometer, capillaries are used for the measurement of viscosity. Foreign organic matter is defined as the pharmacopoeial standard, which is used to control the purity of crude drugs. Foreign organic matter is a material consisting of any of the following. Number one, part of the organ or organs of which from which the drug is obtained, other than the part named in definition. So this means, so we have some parts of the drug mentioned in pharmacopoeial definition. So other than this, the other parts of the same plant, they make the foreign organic matter. In the same way, the molds, number two, molds matter not coming from the source of the plant, it also is or foreign organic matter. And third is molds, insects, and other animal contaminations, they make foreign organic matter. So how we will come to know what is the content of foreign organic matter? So we weigh about 100 to 500 grams of sample, we spread it in a thin layer and clean or a clean white paper. We inspect the sample with unaided eye and also with you by using a magnifying glass lens. So we then pick up those drugs, non-drug portions, foreign organic matter, and we separate them, we weigh them and determine the percentage of foreign organic matter this way. So next parameter is swelling index. So this has limited application because this is applicable for those substances which contain gums and mucilages and have the tendency to swell when treated with water. So here an accurately weighed sample of 1 gram of plant material when placed in 25 ml of glass toppered measuring cylinder. So the internal diameter of the cylinder should be around 16 mm. The length of the graduated portion about 125 mm, and the upward direction, there is swelling. Once you put these substances with water, there is a swelling taking place, so an upward direction, so the increase in the volume, that indicates the swelling index. And the other parameter is RF values. So when we develop thin layer chromatographic plates using specific solvents for substances from natural sources or from isolated or these isolated compounds, the retention factor is how much they travel along with the solvent. So these values obtained in TLC, they are very significant with respect to their composition. So this is another significant value that is used for the physical evaluation of these natural products. So some of the less significant values which are very much specific are fat content, resin content, and the determination of pH. So in the first case, the samples containing wax, fats, others, they are extracted with solvents like ether, light petroleum in a oxalate apparatus for 6 hours. The extract filtered in a pre-weighed flask. The solvent is allowed to evaporate and the residue is dried at 105 degrees Celsius till it attains a constant weight. So the final weight that we get to the weight of the sample that we have taken, so it makes the fat content, because the solvent ether or petroleum ether, it gets this fat out of the sample. Other parameter is resin content. Here the powder sample is refluxed with acetone thrice for 6 hours. The excess of solvent is removed by distillation on water bath. The residue is suspended in water and extracted with solvent ether 3 times with 20 ml. The residue is weighed again and this gives us the resin content. And the last one is determination of pH. So we prepare a 1% and 10% weight by volume solution of the sample and we filter it, check the pH of these filtrates and compare it with standard values that are reported in literature. Just to remind you that these are very, very specific, these parameters which are applicable to resin containing drugs or those drugs which are rich in fat and wax. While as the pH one, it can be generalized one which can be used in many of those samples, natural product samples. Now let's quickly go through the challenges and the scope of the physical evaluation. As we understand now that the physical evaluations are preliminary type of evaluation that are seldom used alone and they lack specificity and they are used only once the other methods of evaluation they are not available. So there are higher chances of interference in the results related to constituents by the related constituents when you follow these physical physical evaluation methods. Most of the physical evaluation methods lack specificity, so newer sophisticated techniques for the determination of physical properties of the medicinal products in presence of interfering metrics they are highly desired. So students, let's now summarize what we have learned in this module. Herbal medicinal products and pharmaceutical excipients derived from nature are checked for their content and purity using different types of tests and assays. Evaluation based on physical property is a significant tool for the quality control and assessment of natural products. Physical properties like moisture content, solubility, melting point, freezing point, extractive value, etc. are employed by all major pharmacopoeias in the quality control of herbal drugs and natural products. Physical evaluation is often used along with other evaluation methods for the assessment of quality of herbal drugs. Progress in natural product analysis is strongly linked to the advancements in the way we are able to determine physical properties of natural products. Thank you.